species. Those are the major. And that's E. coli and Pasteur. That's why when you take an antibiotic, not only it kills out your bad bacteria, it also kills out your E. coli. And that's why we do that. Most of the time when you take antibiotic, that's why it is a good idea to take a pill of multivitamins with antibiotics, unless otherwise it is contraindicated. It's a good idea to take a pill of vitamins. Greg?
Yeah, these PowerPoint slides, are those online also? Can you get to those?
I'm going to put them somewhere as a promise. Yeah, it's kind of on hold one at this time, but I don't.
Alright, yeah, couldn't find it.
But these videos, I got my first link, and I'll have for this one. Any other questions from the previous lecture? Any questions? No? Really? Let me ask you a few and then we'll move on. Rose? Ask me one question that you will make from on the topic of prokaryotes and eukaryotes for the test. Ask me one question that you can ask me on the, let's say you are your teacher, you're making a question on the topic of prokaryotes and eukaryotes for the test. Anybody? Go ahead.
Are all bacteria eukaryotic or prokaryotic?
Okay. Are all bacteria prokaryotic or eukaryotic?
The key difference between prokaryotic and eukaryotic?
What is the key difference between prokaryotes and? Can we answer to that? Presence or absence of? And who has it? Has it? Name three organisms. Name three organisms that are included under the topic of eukaryotes. Fungus, protozoans, and greenhouse. Name two organisms that are included under the topic of prokaryotes. Crystal? Yes, you do. No. Okay. Okay, fine. Bacteria and blue green algae. Those are the only two actually. There are only two types of organisms that are included under the topic of prokaryotes. Everything else is bacteria and cyanobacteria. Everything else is absolutely. They have prokaryotes, they have nucleoid, they have nuclear material, they have DNA, they do not have nucleus. True. Study of, this is a little tricky, so pay attention. I've not told you this, but this is kind of, let's see if you can answer this. Study of bacteria, such as Mycobacterium tuberculosis. That causes TB, tuberculosis. Study of Mycobacterium tuberculosis is called mycosis. True or false? True. Study of bacteria such as Mycobacterium tuberculosis is called bacteriology. Bacteriology. This is a bacterium. Let me repeat my question one more time. Study of bacteria such as Mycobacterium tuberculosis is called bacteriology. I didn't say fungi. That is that is mycology. This just happens to be Mycobacterium. That's it right here. That's mycology. That's my example. But we are going to practice. I'm not going to just throw it at you. I'll practice this with you. One more question, then we'll move on to the new. What else did we talk about? Beneficial rules? Did you do fermentation last time or no fermentation? Yes, okay. All this? Did you go home and do feces farming? Did you check out feces farming? Did you? No? Pasteurization, we did not do pasteurization, right? Okay, let's start the new material. Answer those questions. All right. So last time we did the biofilm code. Today we'll start with detrimental role or undesirable thing that microorganisms do. The very first thing we are going to talk about diseases. Disease. What is the definition? How do you define disease? Disease. The word disease. Anybody? Any abnormality, any malfunction that is away from normality. Okay, good. Acceptable. Anybody else? Disease. Let me just break it down for you. This means away. Ease means comfort. So any condition that is away from comfort, that's disease. When you take my first exam, many of you will walk away with headache. You will. So, disease, would you define that condition disease? Yes. And you have stomach ache after eating food from cafeteria here. Unfortunately. Disease. So those conditions are diseases, but sometimes you like some kind of candy, chocolate, okay? some kind of food, and you eat too much of that, right? Kids do that too. But there are no bacteria involved, right? These diseases are called non-infectious diseases because there are no microorganisms involved. No microorganisms involved in these. People who smoke, many of them, they get lung infections. They get pneumonia, many of them may get, eventually get cancer or other diseases, right? So that is non-infectious pneumonia. If you make food at home in frying pans that are covered with Teflon, right? Teflon non-stick layer, and if you let the pan burn on your stovetop without any grease or without any food, and inhale that Teflon smoke, you can get pneumonia. That's called Teflon pneumonia. It was in 2020, a few years ago. And the company that makes those pans, they are aware of this. It's called Teflon pneumonia. No microorganisms. It's called Teflon pneumonia. Miners who work in mines, coal mines and other things, they inhale a lot of dust. They get pneumonia cold caused by inhalation of coal dust. No microorganism, right? So that is non-infectious pneumonia. But on the other hand, there are many bacteria, fungi, viruses that cause pneumonia too, right? All STDs, okay? Diseases where microorganisms are involved. We talk about bacteria. Mycobacterium tuberculosis, okay? HIV, these are infectious diseases. How about diabetes? Infectious, non-infectious?
Non-infectious.
So infectious diseases are those diseases where microorganisms are involved. Caused by microorganisms. Sometimes, these are misunderstood. Sometimes people think infectious diseases are those diseases that can be transferred from one person to another. Not all infectious diseases are communicable. Okay? Communicable diseases are those diseases that can be transferred from one person to another. Like many STDs are communicable. You can give gonorrhea to another person, but you cannot give Lyme disease to another person. If you have Lyme disease, you cannot give it to another person. If you have pneumonia, you can probably not give it to another, or not all the pneumonias can be communicable, right? If you have rheumatic fever, inflammation of the muscle of the heart caused by streptococcus, you cannot give it to another person, right? So not all infectious diseases are communicable. So bacteria, microorganisms, they cause disease. Another detrimental role of microorganisms, spoilage of food. All of us, we put food in the refrigerator and we forget about it. Days, weeks, some of us, months, and we look out and we think, what was this? Sometimes in the freezer, really. There are bacteria that can grow in the freezer too. Special bacteria, not very many bacteria can grow in the freezer. But I'm talking about spoilage of food at much bigger level. Farmers who do not store their grain, their harvest properly, fungus gets in the silos, and if there's a little bit of moisture there, they will lose all their harvest. So that's what we are talking about. Spoilage of food, okay? Grain harvest by fungi, much bigger level. Billions of metric tons of grain is wasted every year by spoilage of by fungus. So those are two major bad things that microorganisms do. Can you think of anything else? Anything else that you can think of? Anybody? No? Those are the only two things that I think maybe I should ask you. Okay, well, let's move on. Next topic that I'm going to cover is a brief history of microbiology. Some of the major accomplishments that are being made in the history of microbiology. Okay, starting with history of microbiology is as old as the history of mankind, actually. Old edition, hundreds of years back, they could tell the difference between good mold and bad mold. When someone had fever, they grew their own mold actually, good mold and bad mold, so they could tell the difference between good mold and bad mold. If someone had fever, they would pick this green mold, moldy bread, and they would give it to this person, and the person will get better. Really, they had to eat this mold, and they will get better. Similarly, American Indians, if someone had chills and fever, they would go to this tree. No, they would not pray to this tree. They would take the bark from this tree, and they would make the tea. Okay. They would boil, and they would make the tea and give it that the person would drink this tea, and the person will get better. Research shows that the bark contains what? Quinine, which treats what? Malaria. So the history goes way, way back. But the modern history of microbiology started when the gentleman in England made the first compound microscope by the name of Robert Hooke. But unfortunately, he did not see the first life form. He saw many things, but he did not see the person. The credit to observe life does not go to him. He saw many things. He saw a thin sliced pork and other stuff. But someone else beat him before he could see life forms. What is cork? Anyone knows what cork is? Good, dead tree. Piece of dead tree. Yeah, dead tree. Slice of dead tree. Tree was living at one time, but it's actually a piece of slice of stem. At one time it was living. Cork is made up of two. Anyone took botany? So he did not call them cells. The term cells came much later. He simply called these little slices of pork little boxes. We did not call them cells. The term cell came much later. And the cell is what? What is cell? Cell is the fundamental unit of work. We talked about this last time, fundamental unit of life and the cell theory. What is cell theory? It does not differentiate between single cell or multiple cell. It just only says that all living things are either made up of single cell or multiple cell. Anyone knows who saw life for the very first time? Very good. Anton von Leeuwenhoek is the gentleman who first saw life. This is the gentleman, Dutch fellow, from Holland is the one who actually saw life. If you're interested in watching his beautiful diagrams actually that he drew, actual diagrams that are still preserved, Museum of Science in England. Picture is in your textbook on page number 7. Let's look at that picture. Can you see it okay? Oh. This is this from the windhook. This is his microscope. Simple, not compound. Single lens right here. There's a little screw at the tip right here. This is where he placed his specimen. And the specimen, one of the fascinating specimens that he observed, which actually scared the heck out of him, was his own gum material. He took a toothpick and collected his gum material, put it at the tip of this right here specimen placer. And these are the actual diagram actually that are still preserved. Science Museum in London. Right? This is the chain of streptococci that live in our throat and in our mouth. Okay, when you go to the doctor, they tell you you have strep throat. This is what they are telling you, okay? That you have a bacteria that eats your blood. You have normal strep that lives in our mouth, and this is what he drew. But if you have strep throat, you have a pathogenic form of this bacteria that eats your blood, okay? That causes soreness of the throat. Anyways, this is the one that's really scary, this one right here. That looks like a little squiggly worm. This is the normal flora of our mouth that actually moves and keeps our mouth healthy. But he literally fell off from his chair when he saw something moving in his mouth. This is called spirochete. That's the normal flow of our mouth. But later we also found out there's another bacteria that causes what? Syphilis. That also looks like this. That is not supposed to be in our mouth. So these are the diagrams that he drew. He also looked at other structures, other specimens like pond water and other things. But he's the one who first saw matter. Make a note, that's important. So the credit for observing life goes to this gentleman. And time one. When he saw these bacteria, he didn't use the term bacteria or cells. Those terms were not used at that time. He simply called them what? What does that mean? Animal-like structures. The term bacteria or cells, they were not called. Animal-like structures. Animal-like structures. After this life was discovered, the major debate that went on for almost 1 century was the idea of spontaneous generation. 1 century, almost 100 years, the debate went on. The scientists, they were in favor of spontaneous generation. The scientists, they were against spontaneous generation. They did experiments, weird kind of experiments. One of the most, first of all, let me tell you, what is spontaneous generation? Anybody?
The production of organism without the ancestor or...
Okay, I know what you mean. Yeah, life, go ahead.
No, I was going to say the same thing, life from non-living matter.
Good. Life coming from non-living things. That's what it is. Yeah. Let me give you just very simple example. So-called scientists took a piece of cheese, and this is called scientists. Took a piece of cheese, put it in the middle of the barn, and just kind of and waited in the corner for life to take place and fell asleep. And then in the middle of the night, woke up and saw what? A little mouse nibbling on the cheek. Who made the mouse eat? The cheese. Spontaneous generation. That's what they did. So life comes from non-living matter. Cheese is making mouse. And they saw toads, steak. Okay. They are crawling out of the moist soil. So who's making, or maggots, they are crawling out of dead animals. So who's making all these things? Dead animals and moist soil. Spontaneous generation. So that's what spontaneous generation is. Theory of development of living forms from non-living matter. Toads, snakes, mice, flies, born of one cell. So I'll give you a few examples from each side, and then who resolved it once for all and how. Okay. So first fellow who actually rejected it by conducting a very simple experiment was this fellow, Francesco Redi, from Italy, who said, no, there is no such thing like spontaneous generation. And his experiment was very simple. This is his experiment right here. This picture is not in your book. It's from a different book, but the experiment is very simple. What did Francesco Redi did? He took 2 jars, glass jars, and he placed a piece of meat in each jar. And as you can see, he left the mouth of one of the jars open, and he made the other jar airtight by making it airtight by placing a cork or wax seal on it. So no flies, no air could get in. And he left the jars outside his lab for several days, several weeks. What happened? Flies were able to get in, they laid eggs, and maggots appeared. No flies, no air, no maggots. So, no spontaneous generation. It's not the meat that makes maggot, right? No spontaneous generation. End of the story. But those scientists who were in favor of spontaneous generation, they said, no. You have blocked out one of the essential forces that is needed for spontaneous generation. That is air. And he said, no problemo. Okay, let me repeat my experiment. So he, instead of making this jar airtight, he put some gauze. So air was able to get in, but no maggot. No flies. Any difference in results? No. No flies were able to get in. The air was able to get in. Same results. Air is able to get in. No flies, no maggots. Very simple, right? That result. But no. Another half a century, the debate went on. Another fellow jumped in this debate from England by the name of John Needham. He conducted another experiment. He conducted an experiment, and what did he do? He took a flask. He poured some broth in it. This is his experiment, right? Needham, right here. He poured some broth in it. He heated it, sterilized it, left it open in his lab for several weeks. Okay. Now, in his mind, he's proving he's in favor of spontaneous generation. But he proved that the life is coming from where? Broth. Broth is making bacteria and fungi. He's very happy. Spontaneous generation. He is running naked in space. Right, sure. And Swanzani, a guy from Italy, conducted the same experiment, Swanzani, but look what he did to the flask. Sealed the valve. We are at square one. No air, no air. Okay. So the gentleman who dissolved it once for all is who? Louis Pasteur from. Let me show you his experiment. But before I show you his experiment, you may want to go ahead, if you have the notes, let me read you the three major points that he proved. Okay? But let me show you the picture. This is Louis Pasteur. What is this shadow? I think this is light. One of the lights. Yeah. Want to turn this off, please? Thank you very much. Thanks. Okay. So this is Louis Pasteur. The only difference between the other experiments and Louis Pasteur is the shape of his flask. Okay. He also used the same broth, but the shape of the flask was different. He poured the broth in, and then look what he did. He bent the neck of the flask, gooseneck, it's called gooseneck flask, and then he heated. Just like other scientists, he heated. Heat also played a major role. We'll talk about this later as a conclusion of what we found. This is not very realistic kind of flask. I'll show you in a minute. Okay, another picture. Then after heating, he left the flask in his lab for several weeks. By the way, when you're a scientist, when you're trying to conduct an experiment, prove or disprove something, you don't do maybe 5-10 flasks. Hundreds of flasks. Remember, this debate is going on for almost a century. If you visit Pasteur Institute in Paris, okay, there are still some flasks that are still sterile at Pasteur Institute. So, this is what he did. Let me show you. I just told you, talked to you about. Francisco Reddy, rejected. How did he do that? John Needham, supported. Charles Manzani, rejected. Let me read you this. This issue of spontaneous generation, it was resolved, that's the key word, resolved by Louis Pasteur. And there are three major points that you need to remember. And please number them if you want to. Pasteur, he proved what? Number one. Pasteur proved that, number one, microorganisms are indeed present in the air. That's point number one. Let me repeat. What did he prove? That microorganisms are indeed present in the air. That's one. And number two, they means microorganisms that are present in the air, they can contaminate sterile solutions. That's point number two. They, microorganisms that are present in the air, they can contaminate sterile solutions. And point number three. But air itself, point number three, air itself does not give rise to microbial life. That's point number three. Air itself does not give rise to microbial life. Number one, microorganisms are indeed present in the air. Point number two, microorganisms that are present, microorganisms can contaminate sterile solutions. Number three, air itself does not give rise to microbial life. Once we're finished, I'm going to go to the book and show you this experiment. And his experiment is on page number 9. Page number 9. while I'm explaining this to you, try to think this question, try to answer this question. Why all of these scientists, from the very beginning, Francis Corelli, all the way to the Pasteur, why are they using some kind of beef or some kind of broth to prove or disprove their point? Some is using meat, some of them are using broth. Why can't they use water? Well,
It grows faster.
huh?
Will it grow faster?
They will grow faster. Okay. All right, sure. That's one answer.
Do they think they need the nutrients in the food as a key ingredient to grow the?
Okay. Hold on to your answers. Yeah. Hold on to your answers. They can decompose. All right. Hold on to your answer and remind me to ask you this question at the end. Yeah. Because I'm going to explain two terminologies to you that are going to be your exams. Okay. So remind me to ask you this question at the conclusion of this experiment. Okay. Hold on to that. So he also used broth. And as you can see, they are showing you right here in this picture that his broth was also contaminated with bacteria. Right here, as you can see. That's why all the scientists who work against or in favor of spontaneous generation, they heat it. As you can see, they heated their broth to make it sterile at the beginning of the experiment. Okay. When they heat it, okay, the solution becomes sterile. After making it sterile, he left it at the counter for several weeks. What do you think happened when he left his flask on the countertop?
Air will enter.
Air will enter along with the bacteria. Their dust, pollen, other stuff in the air too. So air is going to carry everything and will try to get in the flask. Do you think bacteria will be able to get in or not? So there is no blockage here. See, air along with the dust particle, they are going to try to get in. But because of the gravity, only and only sterile air will get in. Bacteria will travel up, 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 but because of the gravity, they will fall down. That's the trick to this experiment. Because bacteria carry weight. Dust particles carry weight. Okay. Pollens carry weight. They will travel. Okay, that air enter here. They have to travel this distance. Up, 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 up. But they will fall down. All the dust particle and the pollen and everything else, they got trapped right here. And only sterile air was able to get in. After all these years, his broth is still sterile. But not only that, he didn't stop right there. Then after a few weeks, he intentionally, showing you, which is not in your book again, he intentionally took some of his flasks. Look at this flask right here. He took some of his flasks and he tilted them and intentionally contaminated the broth with the debris that was trapped in the neck, and then put the broth back in his lab. What happened? Bacteria was growing in the broth. What did he prove? That air is the carrier of these life. Air itself cannot make life, but air is the carrier of life. That's how he proved there is no such thing like spontaneous generation. Air carries life from one place to another. Air itself cannot make life, but air carries life. End of the story of spontaneous generation. Make sense? First point that he made, broth itself does not make life. Does broth make life? No. Does air make life? Here is the air. Now, once you prove or disprove something, your colleagues around the world, they should be able to duplicate your work, or you are a fake. Like a few years ago, some scientists, they tried to, I think it was in North Korea, this gentleman was claiming cold fusion. Oh, I come up with cold fusion. No one was able to duplicate his work. Right? So, Louis Pasteur also experienced the same problem, that some scientists, they were unable to duplicate his work. Right? Not. So, he had the same dilemma. But a physicist actually, not even a microbiologist, a physicist came to his rescue. He had explanation. Really kind of surprising. Some scientists, they were unable to duplicate his work. Let me tell you how and why. I need to draw the diagram for you to explain this. Any question on spontaneous generation? Experiment next week. Okay. Tuesday, when you go to the lab, you are going to do an experiment in which we are going to use a bacterium called E. coli. Let's say here's a test tube. Another bacterium. We will not be using this bacterium. I'm using this just as an example. Let's suppose we have another bacterium. Bacillus anthracis. This is the one we will be using. This is the one we will not use. But let's say we have another test tube in which we have another bacterium, Bacillus anthracis. Bacteria, when the conditions are good means when they are favorable. Okay. Favorable means when they have plenty of food. Okay. Young cultures. Normally, when the culture is 24-hour-old or younger culture, it is considered a young culture. Any bacterial culture means cluster, group of bacteria. And it is young, 24-hour or younger is considered young culture. Bacteria, they multiply. How do bacteria multiply? By a process called binary fission. One bacteria splits into 2, and 2 to 4, and so on. Binary fission. Let's suppose we leave these two test tubes in the incubator until the end of the term. It is now April. What is it? Ferment. April what? We leave these test tubes in the incubator until April 29. Do you think you will be able to retrieve these bacteria from these test tubes? Now remember, as the number of bacteria goes up, okay, what happened to the nutrition in the test tube? What happened to the metabolic waste? Bacteria also goes to the bathroom. In each test tube there are little bathrooms in there. Not. But they make metabolic waste like acid and alcohol. So the metabolic waste goes up. How about space? Less and less space, right? As the number of bacteria goes up, conditions become adverse for the bacteria to grow. They don't have to wait that long. Within 48 hours, conditions will start to become adverse. Imagine at this stage, any culture, by the way, any culture that is 48 hours is considered old. Is no good. It's old culture. So at this stage right there, imagine, do you think bacteria is living?
No.
Okay. But believe it or not, one of them still can survive. Let me tell you how. E. coli has no mechanism of survival. As soon as the food is gone, E. coli is gone. Has no mechanism. On the other hand, Bacillus anthracis, as soon as the conditions are bad, this bacterium has a mechanism of survival. Instead of multiplying, this is what mommy is going to do. So mommy said, I'm going to save my baby. So instead of multiplying, it's going to make a copy of the DNA. And mommy is going to start making a cement-like structure, and start depositing that cement around the copy of the DNA. So the copy of the DNA is now protected from all the harsh conditions. And this is called endospore. Inside this endospore, you have a copy of the DNA. Or you can call it baby. Little babies inside. Protected from all the harsh conditions. No food, no water, no problem. It's like a seed. Seed. All it needs is a sprinkle of water to germinate. So what happens to mommy? 48 hours, mommy has sacrificed herself. Go on. You boil it, literally boil it. No problem, baby is safe. You zap it with UV light, baby is safe. You freeze it, baby is safe. How long? How long? Forever. Hundreds of years. Hundreds, thousands of years. How can you kill it?
Chemical? Some chemicals? Doesn't some chemical dissolve the spore?
With a little hammer. There are a few things that will destroy it. Number one, autoclave, pressure cooker. That's the most common method. Autoclave, pressure cooker. Number two, ionizing radiation, X-rays and gamma rays, and few types of gases. One of them is ethylene oxide. Ethylene oxide gas. So, now
No
we'll come
acids?
back. Pardon me?
Acids don't work on them? On the spore?
Acid, peracetic acid plus hydrogen peroxide, combination of that. But the problem is you cannot use it on a whole lot of things. It's very corrosive. You can't use it on utensils or not. No. So the problem is we are going to come back to Louis Pasteur's now. Why didn't other scientists who are working with Louis Pasteur's experiment repeat Louis Pasteur's experiment. Okay? When they heated, remember Louis Pasteur's flask, and when they heated their flask, like, right? When Louis Pasteur heated his broth, what do you think? Oh, I did not explain one of the terminology before. All bacteria, when they are metabolically active and they are reproducing, they are called vegetative cells. Vegetative cells. All bacteria, when they are young and they are multiplying, they are called vegetative cells. Okay? All bacteria, as long as they are metabolically active and multiplying, we call them vegetative cells. And when they become metabolically inactive, they are called, oh, one more thing before we go to end. Vegetative cells are heat sensitive. Vegetative cells are heat sensitive. They can be killed by boiling, heat sensitive. They can be killed by UV light. If you zap them with UV light, they will be killed. Endospores, on the other hand, they are heat resistant. Endospores are metabolically inactive and heat resistant. Endospores are metabolically inactive and heat resistant. Now, when Louis Pasteur conducted his experiment, what do you think was present in his flask? What type of cells? Vegetative cells. So when he heated them, they were killed. And when other scientists, they conducted their experiment, what was present in their flask? Endospores. And they survived. They survived. They were not killed. That's why they were unable to repeat the experiment. So John Tyndall, physicist, he's the one who explained that life exists in two forms: bacterial, heat-sensitive vegetative cells, and heat-resistant endospores.
How did he know about the endospores?
How do scientists know anything? Trial and error. Trial and error. The scientist who actually came up with the idea of biogenesis is from Germany by the name of Rudolf Virchow. He is the one who came up with the theory of biogenesis, that life only comes from pre-existing life. That is called biogenesis. Now, other than resolving the matter of spontaneous generation, Louis Pasteur gave us so many other good things, such as pasteurization and fermentation. What is fermentation? Anybody? Fermentation.
Fermenting on the
It's biochemical, biomedical or biochemical pathway by organism to ferment, like alcohol.
Converting alcohol to...
No, by fermentation you produce
Does
alcohol.
it have to be alcohol?
Anything.
Anything like such as
yeast, sugar.
sugar too? Sugar 2 what? No problem, yeah.
Sugar to what?
Does it have to be sugar? Do we have to use sugar?
No.
Is sugar a requirement?
Yes.
As a medium.
Not. Not. I'll give you a very simple definition. Okay. There's several definitions, but organisms prefer sugar. Like, for example, if your body is given glucose and potato, which one your body will prefer to use glucose, right? Of course it can make energy from potatoes too, but your body will prefer what? Glucose. Why?
It's simple.
Yeah, it is simple sugar, right? Yeah, sure. So you can make, fermentation is a process in which, fermentation, the first definition, fermentation, a process in which one organic compound, let me just write it down in a graphic diagram for you so you can actually, make it complicated for you. Sorry. It's time. 15 minutes. You will fill in the blanks, okay? I know you know all the definitions. So we have, first of all, fermentation. Aerobic or anaerobic? Is this process aerobic or anaerobic? Does it need oxygen or it does not need
Anaerobic. Anaerobic.
And without oxygen, it does not need oxygen. And aerobic. Now think of you're making bread. You're baking your bread at home. Do you need to suck out all the oxygen from your kitchen? No. Or your kitchen will blow up? No. So this process is anaerobic. Okay. You don't need to remove oxygen from your kitchen. This does not use oxygen. Okay. It is anaerobic. It does not use oxygen. But it does not require that you must remove oxygen. Okay. So it is anaerobic. It does not use oxygen. Anaerobic, without oxygen. It starts with an organic compound. It starts with one organic compound. It requires a biological catalyst. This biological catalyst, what is another name for this biological catalyst? Microorganism. Microorganism. This converts this organic compound into another organic compound. Now let's look at specific examples now. Let's look at specific examples. Okay. Sugar. Let's take sugar now. Sugar is an organic compound, right? First example. Glucose. Glucose. Bacteria that lives in female vagina. Okay. Its name is yeast. You want to put yeast? Fine. Let's put yeast. I was going to give that example as a piece of bread, but you want to put there? That's fine. No problem. Fine.
Lactic acid.
Lactic acid. Lactic acid. Number 2. Let's make bread now with the same organism. Another organism that lives in the female vagina is Lactobacillus bulgaricus. Lactobacillus. Lactobacillus bulgaricus. That also makes lactic acid. Our so-called friend E. coli that lives in our intestine. Can you see? Or should I use a different color? Glucose, I mean, our so-called friend E. coli. E. coli, it has two enzyme systems. It can do both. It can make acid first. Can make acid. And it can make alcohol and CO2. Yeast, when you use yeast for bread baking, for purpose of baking bread, it also makes alcohol, which gives aroma to the bread, plus CO2, which makes the bread rise. CO2. So yeast can do two things: alcohol and CO2. So here are fermentation, by depending on the biological catalyst, at different end products. And there are other organisms that can make formic acid, butyric acid, acetic acid, and other end products. Okay? But the broad definition, simple definition of fermentation is what? A process which is anaerobic, in which one organic compound is converted into another organic compound by a microorganism. That's it.
I have a question.
Sure.
So, metabolism of glucose, for example, by the muscle,
Yeah.
producing lactic acid, can we call that fermentation in a way?
Absolutely.
Okay.
Yes, it is. Absolutely. Pasteurization. Anyone knows the story behind pasteurization? How did Louis Pasteur come up with that? Go ahead. No, but yeah, that is.
It was wine producer or beer maker or something. They wanted to do it. They wanted their alcohol to last longer than shipping.
Louis Pasteur, France is very famous for exporting their wine, alcoholic beverages, and cheeses to other countries, right? But in his time, Louis Pasteur's time, these exporters, they had a problem. By the time they will reach to the destination, their alcoholic beverages will turn into vinegar. So they went to the Pasteur for help, and they said, please help us save our beverages. Okay, we have this big problem, we are losing money. So he came up with the idea of pasteurization. What is it, pasteurization? Well, we already have the definition. Okay. But pasteurization, is it sterilization? Is it? Pasteurization, sterilization? So let's talk about sterilization. Give me the definition of sterilization, please.
Killing of all microbes.
Pardon me?
Killing of all microbes.
All microbes.
Microorganisms.
Killing of all microorganisms. Is that sterilization? Killing of all microorganisms.
It's complete absence of death, like
All life. Is that sterilization? I have two definitions, killing of all microorganisms, killing of all life. Both are very close, very close. Yeah. But both are not completely accurate. I'll tell you why. Because both of them, they don't include endospores and viruses. Okay? Complete absence of all life forms. That they must include endospores and viruses. If you say all microorganisms, okay, that does not include endospores, that does not include viruses. Okay. Your definition does not include prions and endospores. Okay. So, complete absence of all life forms, metabolically active and inactive. That is sterilization. Sterile. Okay. Complete absence of all life forms, metabolically active and inactive. That is sterile. And pasteurization, it does not kill or destroy or remove all types of microorganisms. It just simply removes certain types of microorganisms. Certain types. By the way, if you have your own cow, okay, you can sit down under the cow and drink milk from the udders of the cow, because you know your cow is healthy, and as long as the milk, okay, that comes out from the clean udders, it's sterile. It gets dirty, contaminated if the udders are dirty, milking equipment is dirty from transportation. The milk itself is a sterile body. Did you know that? Okay. Otherwise, okay, it's pretty clean body. So, I don't know if you have been to the farms and if you've seen the milking process and all of that. That's how all these bacteria they get into the milk. Okay. So, purpose of pasteurization is to increase the shelf life. Okay. Process that kills the bacteria that can be transmitted from the farming process, air. Mycobacteria, salmonella causes food poisoning, mycobacterium tuberculosis causes tuberculosis, tuberculosis of the brain, tuberculosis of the lungs, tuberculosis of the genitals, tuberculosis of the spine. Coxiella burnetii causes what? Q fever. So the person constantly says Q, Q, Q. Not. This bacteria lives or grows in the lymph glands. Lymph glands, they swell up. It's actually toxemia, where the body cannot clean itself. Okay. The Q means unknown fever. But now we know it's caused by Coxiella burnetii, toxemia, blood poisoning. So next time when we meet, we are going to talk about the different types of pasteurization. So today we are going to stop right here. Pasteurization. Have a good weekend.